Most of you, I think, are probably well aware when you get into the general literature around soil health. Very often, the suppression. Can you go ahead and do you want me to do it or?
I will.
Yeah, go ahead. Yeah, the suppression of soil-borne pests, pathogens, and sometimes even weeds are often listed or proposed or promoted as ecological functions of soil that are expected to come from improving soil health. And they're not always talked about like upfront with sequestering carbon, cycling nutrients, structure, aggregation, those sorts of things, but usually the third, fourth, fifth of these ecological functions that people usually talk about. And but it's an interesting area because when it comes to how do you assess that that particular function, it's a little bit more challenging, difficult than for the other ecological functions of the soil that we associate with improving soil health. Fine, we stay there. So most of the other ones, as Charlotte so very eloquently described earlier and with so much detail, most of the other ones, there's been a lot of thought and consideration on how various proxy indicators, things that we can measure, kind of wrap up into giving some indication of the capacity for carbon cycling of nutrients and for sequestering carbon and for developing aggregates and those sorts of things. And so there's been a lot of work that's gone into that and choosing those indicators. And I've just listed a few of them up there. So under sequestering carbon and structure and water and those sorts of things, these are some of the things that we've talked about and Charlotte explained very carefully before. When it comes to the ecological function of suppressing pests and pathogens, it's fundamentally different because it's entirely system specific. With most of the other things, you can kind of translate across cropping systems. That an improvement of soil organic carbon, for example, is important in terms of its indicator value, but also in terms of the functions that it helps to promote are similarly important across all production systems. But every production system has different complexes of soil-borne pests, pathogens, and weeds, and those sorts of things. So it's really difficult to come up with indicators, proxy indicators for measuring these sorts of things. So that's kind of one of the first points I wanted to make about this. But go ahead.
Yeah.
And of course, as Miranda was just talking about, there's all sorts of, the future. We may come to identify or determine ways of using microbial community composition or even the transcriptome composition to find better, additional proxy indicators for all these things. But in particular for that area that I have up there on suppressing pests, diseases, and weeds, I think there's potential in the future to identify or to be able to use some of these microbial community analyses to come up with proxy indicators, but we're not quite there yet. So I'm going to say a few more words about the connection. Oh, that's but the connection between soil health and pest and disease suppression. So this is really an old story. Like it's as old as the history of agriculture, really. So a lot of root pest and disease complexes have long been associated with long-term cultivation and declines in soil organic matter. I mean, plant pathologists have known this for a long time for some of the major diseases of high-value horticultural crops in particular. Not always clear if it's due to monoculturing or due to actually declining soil health, declining soil organic matter, but that association has been there for a long time. And so that's why we associate, now in this modern era, we're talking about improving soil health. We want to assume that we'll also be able to improve conditions with respect to root pathogens, et cetera. Another reason this connection exists is that we know, in the last 75 years, lots of really important soil-borne plant pathology kind of research that most soil-borne pests and pathogens, in fact, all of them, are regulated. Their populations in soil are regulated to some degree or another by other soil organisms, soil antagonists. And then also, more recently, there's been a lot of research on specifically using specific organic amendments, such as compost and various other sorts of things, to actually induce a disease suppressive soil, as a proactive treatment, if you will. So these are all the myriad ways in which we associate the prevalence or the suppression, the inverse of that, of soil-borne pests and pathogens with soil health. So I'm going to flesh out one example that's kind of near and dear to my heart, which is known as apple replant disease complex, just to give you a better sense of how these things fit together. We've known, I shouldn't say we've known, humans have known for well over 100 years that if you try to plant a young apple tree into the site where an old apple tree had been growing previously, that you will not get very good growth. So this has been known for a long time. For at least 75 years, we've known it's biological in nature, that there's something that happens in the soil microbiota in that old apple orchard as it matures that results in it being very inhospitable to those new apple trees grown in there. We've known for 75 years that at least root lesion nematodes are part of that picture. And there are probably some other fungal pathogens and things like that. More recent years, a lot more sophisticated research on identifying a hodgepodge of fungal pathogens and even some bacteria that also contribute to this. This syndrome, for over 100 years, has been known as sick soil syndrome, the opposite of healthy soil, sick soil. We've been talking about soil health in orchards for over 100 years. So the way this works, the populations of these pathogens build up in the soil over time in the rhizospheres of those apple trees. And so then they reach, and so at the levels that they're at on those mature trees, they're not really all that problematic, or at least we don't notice it. They're not killing the trees, but they're definitely influencing their growth. But it's not a huge issue. You pull that tree out, you have those high population densities, put a young tree in, it's trying to grow roots, and it hasn't had time to build up its framework of a stem and things like that. They do not, and so they succumb. We analyze this in a very simple way by using what's called a bioassay. You take soil from an orchard, you pasteurize half of it, don't pasteurize the other half of it, plant an apple seedling, it tells you what the net effect of the soil biota is there. And by and large, usually in an apple replant scenario, you get much better growth in the pasteurized soil than in the non-pasteurized soil, as shown in that picture. Those other pictures are root lesion nematodes, one of my favorite subjects. So from an orchardist perspective, what would you choose as soil health indicators? And I may be going out on a limb here, and maybe Gail, if she's still listening, can chime in, but we have the general indicators of soil health that we have talked about already today. I have at least the three main ones up there: total organic C, mineralizable carbon, and aggregate stability. But you might, from a producer perspective, in terms of what's important to apple growers, you might want to include what I would call specific indicators. A disease bioassay, perhaps root lesion nematode analyses, and perhaps even specific pathogen quantification. And in fact, as part of the replant program that the BC ministry administers with BC fruit growers, there has off and on been a requirement for them to have nematode analyses done to approve their replant. So again, just some of the connections and why it's potentially important. I mentioned that part of the connection relates to the role of natural enemies. I'm going to show you this one bar graph just to illustrate this. And so this experiment was a cherry replant experiment that we were working on. And prior to planting the trees, we fumigated the soil. We had controls, so we didn't do anything. We fumigated the soil, which would be a sort of conventional replant management practice. We applied a compost. And then the fourth treatment was right as the trees were planted, we applied a bark mulch to the top of it. 2 months after, and the colors aren't really clear, they're at least not for me. 2 months after planting those cherry trees, so the left-hand group of bars, the far left-hand bar is the fumigated plots. Very low numbers of root lesion nematodes, which is the main pest of interest in this replant scenario. So that's what those graphs are. All the other treatments, the nematode populations are kind of the same. That's what you wanted to see, because the fumigation works, right? And the other factors didn't really have time to work. You come back 18 months later, so 2 growing seasons later, the largest population densities are in the fumigated plots. So the pest populations ramped up really quickly. The reason being all their natural enemies have been eliminated by the fumigation. They get a foothold, off they go. Then the other treatments, in particular the 2 organic treatments, the compost and the mulch treatments on the far right-hand side, they had lower populations than the fumigated and then the controls as well. That's due because we help to enhance populations of some of the natural enemies of those nematodes. So yeah, there it goes. Okay. So yeah, so soil health management can lead to not only nematode suppression, but this is applicable. I'm talking from a nematology perspective because it's mostly what I work on, but other soil-borne plant pathogen systems, even in annual crops, very similar. I just want to show you some nice pictures here of some of the natural enemies of nematodes that we can document are increased when you're enhancing organic matter inputs to the soil. Top photograph is a nematode that's been parasitized by fungi. Those are spores, little holes in it. Predaceous nematodes that eat other nematodes. Tardigrades, little microinvertebrates that slurp up nematodes. That's a nematode sticking out of its mouth. And the biggest and baddest of all, nematode trapping fungi. These are fungi that live in the soil. And they love it when we add relatively high carbon to nitrogen ratio materials to the soil. It enhances their populations and they literally trap nematodes. Because for them, it's a source of nitrogen. So yeah, so the weird and wonderful world of sort of natural biological control and how soil organic matter management and soil biological activity and diversity relate to pests and pathogens. Left out a couple of examples, but can we assume that soil health improvement leads to pest and disease suppression? You know, sometimes we start saying these things and it starts to become real. So yes, I showed you some examples there. But some amendments can actually increase pest populations in the long term. And this works for fungal pathogens as well as for plant parasitic nematodes that I work on a lot. One example, that bar graph on the bottom right, the exact same experiment I just showed you before about root lesion nematodes and the suppression after 18 months in the compost amended and the rapid growth in the fumigated. So that particular field experiment, we eventually got another nematode in there, known as the ring nematode, that is a parasite of cherry trees, not so much of apple trees, and it's potentially very important. And so you see there, we have the control, fumigation, compost, bark chip, the compost plus bark chip bars on the graph there. Compost treatment, highest population densities after 9 years. One example. Now I have many others from the research that I've done in the past. Some work I did with Shabtai many years ago in his plots in Agassiz on manure application. We found the largest root lesion nematode populations in those field plots in the high nitrogen and the high manure treated plots. So this is not a universal, again, sometimes we look for the good news and we say these things to ourselves all the time, but it doesn't always work that way. Now, cover crops. So I've been talking mostly about amendments as examples. Cover crops are going to be more kind of the universal BMP in all of this. Then you add into that if you're putting cover crops into the system and changing what you're doing, you also have to deal with the fact that there are specific pest pathogen combinations with cover crops. Some of the cover crops may actually be alternate hosts for the pest or pathogen that may be important in your system. So this is really important to keep in mind. The fact that the cover crop may be improving soil health in the long term, how does that relate to the fact that you may be building up pests and pathogens in the short term? So BMPs intended to improve soil carbon and soil health can potentially backfire with respect to pests and pathogens, just depending on your system, depending on the situation. So I think it's worthwhile to look into these things. So some of the work that we've proposed to do with our small little bit in agriculture and agri-food Canada, year one, in a very limited number of sites, crops and sites to be determined yet, the exact ones. And we need to coordinate with Luke's group and with Miranda to make sure we do this to get the most out of the information that we can. We're going to characterize the plant parasitic nematode communities in that first year. Important for baseline, for post-BMP, to compare for post-BMP analyses, and also just for general site characterizations. So for everybody, learning how these sites through the years, how how they proceed. Then in year 4, we'll resample again. We'll compare the nematode communities in BMP and non-BMP fields and plots. And these analyses will include not only the plant parasites, and I didn't even get into this, but free living nematodes are actually quite good indicators themselves of the structure of the soil food web. And there's a whole another little area of analyses that can be done there. We'll use bioassays, take a bioassay approach on the same soil samples to assess the net pathogenicity in those soils. First question, I'm sure people will ask, why don't you just, identify the pathogens that are there and use qPCR probes, coming straight out of like Miranda's type of talk. And again, this comes down to availability of funding, being realistic, what can be done. If practically we can get an assessment, a practical assessment, of the disease potential in those BMP influenced, non-influenced sites using a bioassay, that's the way to go. Because otherwise we won't have any money left over to actually do any analyses if we go with the molecular approach. But it's not out of the question that over the 5 years, maybe through research we're doing in other projects, we might be able to develop something that we could deploy at that time. So, but I don't think we need to plan that, build it in right now. So, and then of course, then we want to interpret the bioassay results in the nematode populations in relation to the other soil health indicators that are going to be measured and the microbial metagenomic data, whatever it ends up being, and to see if we can tease out, fingerprint, if you will, of when we get some disease suppression and when we don't. And most importantly, to compare with crop health and vigor. That is the ultimate take-home co-benefit. We really need to nail that down. So that's not part of this discussion, and I realize that, but I just wanted to remind everybody of that. I think that's going to be important. So yeah, a little bioassay. And this is a picture from one of the biggest cherry orchard programs, I would call it, in cherry orchardists in the Okanagan. There's a relatively new planting of sweet cherries, and see there's compost all up and down the rows, and it was just phenomenal to see that adoption of these BMPs that we've kind of been working on for many years. So it's all an extension, like now into the living lab, of doing exactly this kind of thing. So hopefully you can see this. So I'll stop there.
Thank you. Question. And you do have a first question from Gail Nelson,
Gail.
from the BC Fruit Growers Association. And her question is about money.
Yeah. How much do you have, Gail?
Well, and just to clarify, so you have funding from within AFC to do research, probably focus on tree fruit or wine grape on this piece?
So that's a really good question, because I have, and it's always, these things are really complicated. As you all know, there are cluster projects in the works, agri-science project in the works, and at least a couple of commodity groups. I have projects in the works to take a very similar approach. I would like to most strategically do this work on groups that I'm not covering in those other ones, to try to pull these together.
Okay.
And without getting too far into the details, that's one of the considerations.
So all these clusters came through.
Yes.
What crops would you be covering?
Probably, probably apple and small fruit. Yeah.
Okay.
Probably. Maybe grape. Well,
Okay.
yeah.
That's really helpful.
And so there's that element, but also to add to that complication though, is that, apple will be there for sure. To add to it though, it depends on, because there's going to be some strategic planning in terms of what analysis Miranda is going to be doing on which systems, and likewise Luke's program.
Right.
Because I would really like to be able to do this somehow in concert with them. So at least we're looking at the same fields when this is done. So it's going to be a little bit tricky to just pin down exactly. Yeah.
Great. Okay, well that's helpful.
We'll get there. Yeah.
And so Gail, her question was around their budget. So this is coming from AFC's own budget for this project.
Yes.
So you don't need the budget for Gail. That would be the short answer.
Yes.
But that's really helpful because I feel like other people would ask the question, which crop?
Yeah.
Is that also like funding from the Living Lab program or from other programs?
It's AAFC's component of Living Lab program.
Okay, now that we started the questions, who else has questions? Yeah, go ahead.
I was just wondering, you shared some interesting soil related pathogen sort of responses to soil related practices. Is there any information about how that translates to above ground pests? Does the soil community translate through the insect population up into the above ground?
Yeah. But actually, that's a really hot topic of research. And, I'm aware of research projects and papers. Even foliar fungal pathogens can be influenced. But these are still, these interactions, sometimes I wonder about their robustness in the real world. Often these interactions are being studied in plants in low growth chambers. The potential is there, very definitely. Things happening on the roots can influence and the way the roots are interacting with rhizosphere microbes as well as soil invertebrates. Very definitely can influence through signaling in the plant and influence susceptibility to leaf-eating insects, powdery mildew, those sorts of things. But demonstrating these things happen there and in the field, are you going to be able to observe a one-to-one connection? It's not to say it's not happening, but there are all sorts of other things that are affecting it too. So but it is because that's that's research that kind of adds to our general understanding of that.
Other questions? Okay, I'm gonna throw another question. I'm gonna go using my powers as moderator. Are there other researchers within AFC that might be doing similar research on other cropping systems? So berries, perhaps, or field vegetables?
Good question.
It's okay if you don't have the answer. I just that I would ask.
Well, in terms of similar, so
Similar in that they're looking at the pest soil.
yeah. So Richie Burlico
Oh, okay.
is a plant pathologist at the Aggie Research.
Mhm.
And I'm not sure if he was even part of these discussions early on or at what point he would have you, but he's doing work on Phytophthora root rot in blueberry and raspberry for sure. And that could be,
Mhm.
a very similar.
Basic.
Yeah, exactly. And in the Okanagan, so Jose Urbis Torres, our plant pathologist up there, I actually work with him, collaborate with him on some of the stuff we're doing on grape. And likewise, he's done some of the analyses of fungal pathogens associated with cherry tree plants, for example. But again, this most of these researchers, I might be speaking out of line, but the amount of money that's available to do this is, bring others in. It's not to say that I don't want to share. That's not it, but it's just like, everybody's like, well, how do we do this?
Yeah. And maybe over the 4 years remaining, there could be opportunity for other funding that could be like sort of built on.
Yeah.
Actually. Or maybe not.
It's hard to say because I can't speak for Rishi. Like, I don't know exactly what proposals he has in. Well, I have some idea, but what proposals he has in with the small group, for example, for the next 5 years. And so what he's planning to do with them, what he's planning to do could resonate with or overlap with some of this kind of stuff, soil health related side of it. I don't think it is, but it could. But we get into these 5-year cycles of funding, and then there's not a lot of.
Right.
Yeah.
I see what you mean.
Yeah.
Yeah, so we're at the beginning of the 5 years, so everything is gonna be set.
Yeah.
Okay. Are there other questions?
I'm usually on my quota too.
It's kind of like we are surprised we just ask a question. What about for our virtual attendees? You guys can also ask questions. You can use the chat or you can unmute.
Yeah, just very briefly, if I may. Thanks, Tom, for that presentation. Very interesting. But to the question of above ground impact of the microbiome, I do actually believe that depending on what kind of a cover crop regime you put in place, especially if you let your cover crop grow without mowing for a very long time, I do actually believe that you establish an above ground sort of a microbiome ecosystem. That would also be really interesting to follow, to see what impact it does have on some of the pest fungi potentially. I think there has been some, maybe it's coincidence, but some experience that seems to indicate that cover crops, unmanaged cover crops, can sort of reduce the impact of powdery mildew on certain crops. And we've seen this both in grapes and in apples as well, in the Okanagan and the Similkameen. Would be interesting to actually, I don't know whether anybody is looking at this or whether this is a field of interest for any of the researchers.
Yeah, Hans, yeah, I know, we've talked about that before, and I agree. There's a phenomenon there that there's been some anecdotal evidence that is worth pursuing and following up on. And the living lab experiments could very definitely be a really great platform for that once they get up and running. I don't know if we have anybody in place lined up. I remember in the very early stages talking with Jose about this, or Jose was part of the conversation. I don't know if he's still part of that conversation long term, but we had some of this co-benefit stuff lined up for just a little bit of assessment in the last year. And so there may be some potential to get Jose online or something like that to, by the 5th year to go out to where these BMPs have been in place and to actually do some quantitative assessments. But certainly, like to really pursue it scientifically and to actually characterize those microbial communities on the leaf surfaces, those sorts of things. Now we're into a whole nother
Yeah.
huge area. Yeah.